Good morning, and good evening. I don't know where you are. Today is a real pleasure for me to having with us Pau Sancho-Bru. He's a group leader. He works at the IDIBAPS in Barcelona. He's an associate professor also of the University of Barcelona. His group is conducting translational research mainly due to the location of the institute that is surrounded by the university, a clinic. And his work is in the field of liver diseases. He's investigating the role of cell plasticity in the wound healing and cancer. And one of the aims of his research group is assessing the potential of stem cells for biomedical and biotechnological applications, and particularly to develop a 3D organotypic in vitro systems for disease modeling and drug development. Pau is an old friend of us. I'm really happy to have him here. He's a very talented scientist, and his track records of publications can speak by themselves. But I can tell you that Pau is, regardless his big talent, he's a good person to collaborate. So please enjoy this opportunity to interact with him and to learn from his work. So Pau, thank you very much for accepting our invitation, and the floor is yours. Thank you very much.
Okay, well, thank you, Natalia, for the kind introduction and also for the invitation to be here with you today. So yeah, we in the lab we have different lines of research. We work, I mean, as you said, we like to develop in vitro models, and for that we work on liver organoids and things like that. But today I'm going to focus on our work on IPS and trying and how we use them to understand, or to try to understand, fibrosis. So let me share my screen. Okay. Oh, okay. So do you see it well?
Yes.
Okay.
Yes.
So okay, so basically is what I said, and I want to start talking about the title and the trajectory. I mean, you've been probably hearing about trajectories coming from the single cell studies, in which they develop the trajectories, how the cells evolved. But we are for this topic we are going to use it in another way. So you can imagine that when we developed and we are children, adults, and we get old, this is our physiological trajectory, and at a certain moment or our liver, or we in general, this trajectory may change, and can go into disease trajectory. And we think that this is also true for cells, and IPS are probably a good example, So induced pluripotent stem cells, they are embryonic like cells, and they differentiate into adult cells. So they come, they do a journey that it can be or not related to the embryonic development, but they evolve from undifferentiated to progenitors to adult cells. And if you have oops, sorry, do you see it still?
Yes, we can see it.
Okay, because I just now I just pushed the I'm just trying to minimize all the screen. Okay, no, wait.
But we can see it.
Yeah, but I don't. Now.
You don't. Okay.
It's just all the people just appear here. Okay, now perfect. So as you can imagine, this project is built on the idea that if we understand this trajectory and all the regulatory elements that participate in cell fate and cell identity, we will be able to understand when this trajectory changes and go into disease. And we think that these factors might be also important for pathogenesis. Okay, so as I said, we are going to talk about hepatic stellate cells and fibrosis. You know that fibrosis is the common result for most chronic liver diseases. No, it's linked and strongly associated with disease progression, but also is one of the main factors that determine morbidity and mortality in these patients. So just to show the relevance of fibrosis itself. So you know that the main cells responsible for fibrosis are hepatic stellate cells. These cells reside here at the space of Tisse, lining the hepatocytes and the endothelial LSEG cells. And these cells participate in the homeostasis of this extracellular matrix, the very soft extracellular matrix of the liver, and participate also in the control of metabolism of the liver, the physiological homeostasis. But when there's an injury, they activate and transform into a myofibroblastic cells. And here you can see some of the hallmarks of the hepatic stellate cells when they activate. So cells in a healthy liver, they are in a quiescent state and they activate during fibrosis. And if this injury persists, is when you have the accumulation of extracellular matrix and fibrosis, etc. So these cells start to proliferate. And they become inflammatory, they move, they become more contractile, and also they change their bioenergetics. So there's an important metabolic changes in these cells, basically, most probably, to accommodate their energetic demands and to be able to really fulfill all these new processes that take place during the activation and during the fibrosis, in which they are much more active than in the quiescent phenotype. Okay, so this is how they look in vitro when you plate them from human and they activate in plastic. So this is the most common way of activating the cells, and is how we usually work with them. I mean, I'm just putting this figure because I'm going to talk a lot about activated and quiescent and also in vitro activation. So this is how they look, no? So the cells start to acquire these myofibroblastic characteristics, and in this phenotype, they more or less recapitulate on plastic. They recapitulate the characteristics of the myofibroblasts when they are in fibrosis. So as you can imagine, I've been working with for many years with primary cells, but it's kind of tough to really obtain them. It's you need patient donors. It's difficult. They have I mean, it's limited the amount of cells. That's why people is using cell lines, etc. But I mean, at a certain moment, we thought that it was important to use iPS or stem cells to try to obtain these cells. And we worked on iPSCs, on induced pluripotent stem cells, basically because they have the potential to differentiate to any other cell type. So that allowed us also to generate other cells of the liver, which I'm not going to talk about today, but we could also differentiate them to hepatocytes, stellate cells, inflammatory cells, etc. And also we can obtain them even from patients, but it's a renewable source, so we have as many as we want. So based on these premises, we started working on trying to differentiate them. As I said, induced pluripotent stem cells have an embryonic phenotype, so they are embryonic-like cells. So what people tries to do when differentiating them is to try to mimic the embryonic development of the cells that they want to differentiate. In this case, hepatic stellate cells. So there's not that much known about the embryonic development of stellate cells. So, but we tried to mimic that by developing an in vitro model of an in vitro developmental procedure to try to bring cells from pluripotent state to a mature phenotype. And you will see how some of these markers will appear now when we do the characterization. No, so in these 12 days, cells progressively change the phenotype and acquire the mature phenotype. And basically, we monitor that by the expression of PDGF receptor beta and also by the ability of accumulating vitamin A in the cytoplasm. And you can see how the markers that are typically expressed in mature, but also in more mature stellate cells start to be expressed. Sorry. Along the differentiation, along the 12 days of differentiation. No, you can see how PDGF receptor beta goes up, LRAT, PCDH7, relin. So all these markers that have been described in primary cells also happens in iPS derived. And here I just show you some pictures taken at day 4, at day 8, of markers that have been shown to be expressed in progenitor populations of stellate cells, such as, for example, mesothelial or submesothelial cells. So, I mean, as we do not claim that we really recapitulate all the aspects of the embryonic development, what we see is that they differentiate and they become like first progenitors and then more mature, acquiring a more mature phenotype. So this is how they look at day 12. You can see this is directly differentiated cells, and you can see how they express most of the markers that are expressed also in primary cells. Maybe what is more different is the expression of, for example, here bimentin or alpha-smooth muscle actin. So you can these are two markers that are typically expressed in more activated cells, more myofibroblastic, more fibrotic cells, and this is why and this is because the iPS differentiated cells are in an intermediate phenotype. They are not totally quiescent, but they are more quiescent than the primary cells or the cell lines that we usually take. In fact, when we put these differentiated hepatic stellate cells in culture here in the middle, you can see how these markers of activation resemble much more the ones that we have in the primary cells that we have in culture. Remember that picture, no? That I showed you. Those are the cells that are here stained at the bottom. Okay, so we showed that these iPS-derived stellate cells can be generated. We perform a very broad characterization regarding transcriptomic, phenotypic, functional characterization of these cells to show that they are similar to primary hepatic stellate cells, and we also show that by themselves, by modulating the activation on plastic or TGF-beta, or in combination with hepatocytes, we can generate spheroids and we can mimic fibrosis models or toxicity. Okay, so we are quite convinced that the protocol it's robust enough to give us information that is relevant for the disease. That's why we have started to try to understand this differentiation and to try to obtain that information that we think it's important to understand the trajectory in health and the trajectory in disease. Okay, so here I show you some works that we are doing at this moment. We are profiling the cells at different days of the differentiation by single cell. And you can see how along the days of the differentiation, the stellate cells start or the cultures start to become more complex. Different populations of stellate cells arise, and also the communication between the different cells that form this heterogeneous population that we have in culture when we differentiate the cells start to talk each other. No. If we look on the main stellate cell compartment that we have at day 12, I have to say that about between 70 and 80%, depending on the differentiation, they express markers of hepatic stellate cells, PDGF receptor beta, etc. So by single cell transcriptomics, we have also more or less the same amount of cells that we can attribute a gene expression profile of hepatic stellate cells. So within these populations, we have defined different clusters. So the first one is the stellate cell one, is those that we call remodeling. So they are similar to the ones that have been described also in human samples as mid-lobular like stellate cells. Then there's a proliferative population that is very similar to remodeling but proliferating, and we are still analyzing them to be sure the exact translation to the human tissue. We have stellate cell 3, which are the more quiescent. So within all of them, is the population that shows more quiescent phenotype, but also with some characteristic of the angiogenic profile that has been described in some populations of hepatic stellate cells. And finally, we have another which is more activated, as we were expecting. No, some of the cells that are in this differentiation show more myofibroblastic cells characteristics, and these are some of the markers that we use to characterize them. For us, maybe the more interesting ones are the ones that are remodeling. So these cells express markers of what we understand as activated, but they lack the expression of alpha smooth muscle actin. They lack the expression of high levels of collagens, etc. They produce extracellular matrix, and this is something that we expect from a functional hepatic stellate cells. So just to show you an example, this is a single cell transcriptomic data from healthy liver, and these are the stellate cell 1, which correlate into the cluster. It correlates also into one of the clusters that are defined in this human. So in this human data. So giving us the information that there's a correlation between the populations that we are obtaining and the different populations of stellate cells that are present in a healthy or also in an activated or in a fibrotic liver. Okay, so another approach that we are following in trying to understand this differentiation. Okay, no, I just missed one. So, well, okay, just to show you. Okay, so I think we have enough information right now I forgot to mention that one of the important findings in the single cell analysis is the finding of intermediate populations of this differentiation that more or less resemble, although we don't have single cell data from the embryo that is that clear at the same stages, but they resemble also intermediate differentiation developmental stellate cells. So we are kind of confident that by doing the differentiation, we recapitulate more or less the development, and when we activate them in plastic, we recapitulate this disease stage. So based on that, we started a story in trying to characterize which could be the drivers that can drive the cells in this trajectory. And for that, we perform a proteomic analysis at different time points of the differentiation and also during the activation. So these are the results of the proteomic analysis. You can see how, as expected, no? If you compare day 0 and day 12, there's an important number of cells related to collagen metabolism, extracellular matrix, TGF-beta, wound healing. So no, all the cells all the markers that we expect to have in hepatic stellate cells become expressed and are repressed, all those proteins related to pluripotency, cell proliferation, translation, etc. No? So this is the typical pattern when you differentiate the cells. But what was interesting is when we took all the different samples from the different days, from day 0 to day 12, and we perform an unsupervised clustering, we saw that the cells group into 3 main phases. No, so one phase that is related to mesoderm induction, a second phase that is related to the specification of more mature or more immature, but already pointing into the direction of stellate cells, and a third phase that is where most of the markers typically expressed in mature hepatic stellate cells become more expressed. So these 3 main. So one of the questions that we asked ourselves was, okay, so which are the drivers that might be transitioning the cells from phase 1 to phase 2 and from phase 2 to phase 3? I have to say that these 3 phases more or less are similar to the days in which we change, no, the different growth factors that we add at the differentiation. Okay, so to try to understand how the cells transition from phase 1 to phase 2, for example, or 2 to 3, we analyze the transcription factors that could be involved in this transition. And as you can see, several transcription factors appear. But I just want you to focus on RORA alpha, which was one of the transcription factors that was highly predicted to be involved in the phase 1 to phase 2. Another thing that we performed was the direct comparison between the differentiated cells and primary cells. So we could see that there was a significant number of proteins that were common in both primary and differentiated, but not all of them. No, you can see here in a PCA representation how the different cells, from day 0 to day 12, the cells progress along the differentiation, but they still have this difference between primary cells. So in this dimension they are quite close, but in this dimension, which is already 16%, no, they are not the same. So we also asked to ask which could be the transcription factors involved in these differences. And as you and here are some of the results. So some of them are very interesting, but we were surprised to see again the RORA transcription factor that appeared here. And we decided to focus on RORA because when we compared the transcriptomic profile of quiescent and activated cells to our proteomic analysis, the RORA transcription factor was the only transcription factor that was predicted, that was missing, no, in these differentiatedly expressed genes, and was predicted to be necessary to be expressed in our cells. So if you look at the expression of RORA along the days, you can see how RORA becomes expressed at day 4. So when there's the entrance to this from phase 1 to phase 2, and then gradually goes down and is weighed down the primary hepatic stellate cells. So this was telling us that RORA could have an effect during the first step from phase 1 to phase 2, and also during the maturation phase. So that could have 2 important moments during the trajectory of the stellate cells in which RORA could be relevant. Okay, so RORA is the retinoic acid related orphan receptor. So it's not known who is binding to RORA, but it's been described to be involved in many processes, from developmental to disease. So, and if you look at most of the papers or the bibliography, it's related to mesodermal or mesenchymal organs. In the liver, there's not that much information, but it's been involved to play a role in also in the metabolism of hepatocytes, and also in NAFLD. Okay, so to try to understand which could be the role of RORA, we decided to look at two different moments. No, one during the differentiation, and also during the activation. So you can see, so basically what we at the beginning, what we did is to treat the cells with an agonist of RORA. So during the whole differentiation, well, not the whole, I think we started at day 2. And what we saw is that by doing that, the expression of markers involved in or related or markers of specific markers of hepatic stellate cells were highly increased, and we could see that by gene expression, by PCR, or by transcriptomic analysis. No, so all the markers of mesenchymal stellate cells, and also the pure markers of stellate cells were increased. In fact, in this PCA, you can see how there's a big difference between the population that was not the treated and the not treated cells. On the contrary, when we used an antagonist to and we blocked the expression of RORA with an antagonist, we saw that the cells do not differentiate well. No, as you can see how the expression of typical markers of hepatic stellate cells go down at the end of the differentiation, and also these cells look more fibrogenic, no, more activated. No, there's an increased expression of collagen logs, and also here with Sirius Red, we also saw it in culture. We generated a knockout, IPS knockout line of RORA, and with, I mean, we generated an homozygous knockout, and what we saw is that the cells die at day 4. So as soon as they enter the mesoderm commitment, they die. So we had to generate an heterozygous cell line, because otherwise we were not getting results. I mean, some cells survived, but they were not stellate cells anymore. So when we used these heterozygous lines, what we saw is more or less the same as when we used the antagonist. There was a reduction of the expression of stellate cell markers, and this reduction could be more or less rescued by using an agonist of RORA. No, here you can see how more or less is recovered. Okay, so with this data, we were more or less convinced that RORA seemed to be necessary along the differentiation. So it was necessary to really acquire what we call an hepatic stellate cell phenotype. Okay, but what about their role in activation? No, so what it happens with the stellate cell when activates. It happens that their transcriptomic profile changes completely. So and some of the characteristics of the quiescent cell get lost and disappear. No, others have described how some transcription factors involved in quiescence disappear. No, when they are activated, and we were wondering if this factor that is involved in commitment of the stellate cells could be also disappearing during activation. So what we perform is a very simple activation phenotype. We more or less we did all this with TGF beta, but I'm not going to show it, but because the results were more or less the same. So, but we're more dramatic when we put them on plastic. No, as I said, this is the classical way of activating them. So we put this RORA knockout or heterozygous cell line in plastic, and you can see how the activation of these cells, which was much higher. You can see just by regular eye, but you can see it also at transcriptomic level. Also, we treat these cells during passage with the agonist in green, and you can see how the more quiescent markers such as LX2 or PCDH7 or LRAT were increased, and when we used the antagonist, they were even further reduced, and the activation markers such as collagen, ACT2, or LOX were increased. So meaning that we could even play with the agonist and antagonist on these cells. We perform to try to understand what was doing the RORA in the cells. We perform a transcriptomic analysis. And we could see, as you can imagine, we could see how the cells change and become more activated. But one of the things that we found it could be more interesting was that the bioenergetic of the cells, the metabolism of the cells, seemed to be changed. And as I said, this is one of what we consider a hallmark of stellate cells, because it is required this change, this more glycolytic profile of the stellate cells to have and to persist in their functions in activation and fibrosis. So there's quite a lot of bibliography and about the bioenergetic of stellate cells. So, but I mean, it's not really my subject. So I'm just gonna show you very briefly some of the results that we got. We have also lipidomics on that, but it's too complicated for me. So basically, when a cell starts the activation, they need to change from oxphos to more glycolytic profile to generate energy, and also the lipid metabolism completely change. So this is what we checked in vitro. We use a seahorse, and we treat. I mean, what we did is to passage the cells. And then treat the cells with the agonist of RORA. And what we saw is that this shift into more glycolytic profile was reduced. Was reduced when we treat the cells with the agonist of RORA. And this was during the passage, which is already known that the cells need to change this metabolic rewiring during activation. But we also wanted to check if this was also happening at the early time points, when RORA also seemed to play a role during the differentiation. So this is what we did. We analyzed the cells at day 4 of the differentiation. When if you remember, is the moment in which RORA plays a role and the cells enter into the mesoderm. At that moment, we didn't know anything about that, but it seems to be that mesoderm needs to shut down the glycolytic profile of the embryonic stem cells to be so the embryonic stem cells need to shut down the glycolytic profile that they usually have to be able to enter into the mesoderm differentiation. And this is what we saw. I mean, the results were not that clear here. We couldn't use for the setup of the experiment. We couldn't use the same approach. We use the heterozygous RORA cells, and we treat them with the agonist to rescue. So what we could see is that the glycolytic profile was even higher, when we take out RORA, and it was slightly reduced, not completely, but it was slightly reduced when we treat the cells with the RORA agonist. This was more clear if we look into the gene expression profile of those markers that are important for mesoderm induction, such as EOMs or vimentin. And here you can see the staining. At day 4, RORA cow cells do not express EOMs or vimentin, and this can be rescued when you treat them with RORA. To see if this was something that was happening only in our differentiation, and not in other mesodermal differentiations, we perform a general mesodermal differentiation protocol with iPSCs. And we also saw the same profile, that the induction of a general mesoderm, not specific to stellate cells, was also blocked or reduced with RORA knockout. And we also saw that when we looked at other mesoderms, such as, for example, the differentiation to kidney cells. So with these results, we showed, it suggests that RORA plays a role in regulating metabolism during the induction and differentiation of the cells, and also during activation. You need to reduce RORA to be able to activate and change the metabolism into this more glycolytic and more fibrogenic profile. Okay, so to try to see that these are cell lines in vitro. It's human information, because those are human iPSCs. But we wanted to see the effect in a more complex system. So we went to the mice, and these are the results using staggered mice. These are knockout RORA alpha animals spontaneously that were generated a long time ago and have been used for many studies. So when you induce a fibrogenic insult with CCL4 during 4 weeks in these animals, you can see how they show an increased deposition of collagen, and also an increased activation of hepatic stellate cells. So we generated LRAT-Cre RORA knockout cells. Because, as I said, RORA also plays a role in the metabolism of hepatocytes and many other cells. So we wanted to look specifically in stellate cells. So and you can see how when only stellate cells miss or don't express RORA, you can see how there's an increase, induction of fibrosis, more activation of hepatic stellate cells, while the infiltration and the inflammatory response was not that affected. So to try to go into more translational and see if we could see if the agonist could play a role also in fibrosis. You can see here, this is also CCL4 in wild-type mice, and we treat them with the agonist of RORA, and you can see how by treating the animals, there's a reduction also in collagen deposition and alpha-smooth muscle actin. And we thought that since it is affecting the mesoderm and also the activation, and we thought that this could be also happening in other organs that are affected with fibrosis. That's why we perform also a study on the heart and also the kidney. And you can see how the results were more or less comparable. So we had a reduction on the amount of collagen that is being deposited, and also a reduction in the activated of pericytes or mesenchymal populations in these different animal models. So to try to see that this was something that could be happening also in human disease, we performed just some stainings and some correlation studies, and you can see how in healthy humans you have high levels of expression of RORA, while this expression is reduced in cirrhosis. Here I missed, I think the green one is alpha smooth muscle actin, and the red one is probably vitamin A. I'm not sure about that. Well, anyway, when you look specifically at the gene expression level of activated, so cirrhotic stellate cells isolated from cirrhotic livers and compared to healthy, you can see how there's a reduction. So general gene expression level of RORA correlates with fibrosis and also with the expression of markers of typical markers of activation of stellate cells. So showing that also in liver fibrosis there's this reduction in RORA. Okay, just to finish, basically what we think is that or what we think that we are showing is two things. I mean, it's nice we have identified RORA playing a role at different moments, no, at development and also during activation. We are not sure what is specifically regulating RORA, but we know that it's regulating the metabolism of the cells, and regulating this metabolism is important not only for the commitment, no, the entrance to the mesoderm, but also to the activation of hepatic stellate cells and the induction of fibrosis. But what it likes for us, besides that, is that we think that or that we can learn from this is that we can use the trajectory of the IPS differentiated stellate cells from the differentiation to the activation and the models of fibrosis to try to identify those factors that are important for the identity of the cells, because those factors most probably will be lost or changed during the activation. And we think that by targeting these identity factors might be good strategies to reduce the activation or to revert the activation of hepatic stellate cells. Well, just to finish, just to mention the people in my group, that this is work from Raquel and also King, Maria, and Laura that are working on the IPS work. And well, thank you very much for your attention. That's it.